So good morning, good afternoon, good evening, wherever you are in the world. This, as was mentioned, is the third of this webinar series, and it's a change in track. It's about cheminformatics and computational chemistry. And it's about the virtual screening of the fragment screening hits that have been generated at the Diamond Light source at Harwell in the UK. I'll explain more about this as we go on. Now, in the first webinar, we had an overview of the virus genome. This is just a recap of that. As you'll recall, it's a 30 kilobase pair RNA genome, and there are a number of polyproteins created, and those polyproteins encode a number of functional proteins that are necessary for the virus. And just a quick casual look at this identifies a number of very obviously druggable targets, or targets that should be druggable, one might think. This is not an extensive analysis of this, but a very casual analysis, just immediately for at least four targets pop out. There's an RNA-dependent RNA polymerase, there's a papain-like protease, and a main protease, which is the topic of today's webinar. Those two proteases are responsible for cleaving out the proteins from the polyprotein. And so clearly, if you can inhibit those proteases, you're going to have a very big impact on the virus. As I said, the main, the target of this webinar is the main protease. It's also known as the chymotrypsin-like protease, sometimes referred to as 3CL-Pro. Although it's a chymotrypsin-like, it is actually a cysteine protease. The cysteine is at the catalytic site, but obviously serine and cysteine differ only by one atom. And that enzyme is incredibly similar to the equivalent enzyme in the SARS virus, which had already been studied to a significant extent. And there'd been some fairly extensive efforts to create some inhibitors of that enzyme, but to date, I don't believe any molecules have been discovered that were potent enough to be therapeutics. There's also a lot of mechanistic analysis on that on that enzyme that can almost certainly be transferred straight across to the SARS-CoV-2 enzyme. There's a very good summary of this in the PhD thesis that's mentioned here at the bottom. References here are all in the slide notes, and I believe the slides will be distributed after the meeting. So it's a cysteine protease. So some of you probably already cottoned onto the fact that we could potentially make covalent inhibitors to this enzyme as well as non-covalent ones, as the thiol is somewhat reactive. But this webinar, the information we're describing in this webinar, is purely going to describe the design of non-covalent inhibitors that are based off the fragment screening hits that I'm going to describe. But this work is part of a much bigger effort that's going on, that's been referred to as the Moonshot project. That you can find at this URL, and that project, that broader effort, most definitely includes the design of covalent inhibitors as well as non-covalent ones. And it's a really good example of crowdsourced science. There are a huge number of people who are coming together to contribute to that effort. So, what is a fragment screen, and what are fragment hits? So, the process really has been running at breakneck speed. As we all know, the virus has only been known for a very short time. The genome was synthesized late last year. At Diamond, they synthesized, so they purchased the synthetic gene for the main protease. They expressed it. That happened on February the 14th. By less than a week after that, the first structure, protein crystal structure, had been solved at 1.25 angstroms, and the fragment screen started a few days later. The day after it started, we had some fragment hits coming from that screen, and then just over a week after that, the first 7 of those crystal structures were made available to the public, and then a week or 2 after that, that number had gone up to 78. This is science going on at a breathtaking speed. I could hear you all virtually gasping at this. It is quite an amazing bit of work here. And the work that we're describing, which is the virtual screening going on the Galaxy platform, really began very early in March, around about March the 4th, and so sort of overlapped with that procedure. And so what fragment screening is, which we'll describe a bit more on the next slide, we've got some pictures here. Well, first on the top here is the synchrotron at Harwell. This is the diamond light source. You start with some crystals of the protease, and you can then solve for that structure using the X-rays in the synchrotron. You can solve them very quickly and a very high resolution structure. But what in particular is fragment screening? Well, it's a process where, well, in the old days, crystallography was a bit like a cottage industry. It was done with loving care and took a long time, and you had to be very highly skilled to do it, and it took a long time. Now, with the work they've done at the Diamond Light Source, they've basically put in a lot of automation on this, and they can do this in a much higher throughput basis. What you essentially do is you take a protein crystal, put it in a very small droplet of liquid that contains what we call a fragment, which is a very small molecule that could potentially bind into the to the protein. These fragments are much smaller than typical drugs. They bind much more weakly than typical drugs. So they're not drugs, but what they can do is give you very good insight into how you might want to design drugs, because those fragments give you very good information about how a molecule can bind into in this case, the protease active site. And what fragment screening is basically to do this on a large scale. So you can do this on a library of compounds. The typical library they use at Diamond is the poised library, and it contains 768 compounds. They can process those molecules in mass, put them into the crystals, put those crystals into the beamline using the robotics and the automation that's sort of indicated in the pictures in the top right corner here. And you can do this process with sort of tens or hundreds of these a day, and it becomes a sort of a medium throughput activity. It's quite awesome to see this machinery. And so out of this, as we saw in a couple of slides earlier, we had an initial set of fragment screening hits. So each of those is a protein structure, let's say represented as a PDB file, and in that protein there is the ligand, the fragment hit that is bound into the protein. We're only considering active site binders here. There's some fragments bind to other regions of the molecule, but for obvious reasons, we're concentrating on the active site here. So basically, the virtual screening that I'm going to describe has evolved significantly from early March when we started it. Initially, we had 17 fragment hit structures. So that's 17 different crystal structures, each with a different fragment bound into it. So the workflow has been taken place in 3 steps in a number of phases. The first phase, which is complete and essentially what we're describing here, was a stage when we were trying to get everything done as quickly as possible. It was running at breakneck speed. Once that was over, we sort of took a step back and have been trying to work out how to do things a bit better. So firstly, phase 2, we started to try to improve the Galaxy workflow itself, make it more efficient. And now we're working on actually improving the workflow itself, the steps in the workflow, to make them scientifically more rigorous and better. The main lesson I guess we learned from this is that if you're going to perform a large-scale project like this, you really ought to have your workflow fully optimized and validated before you do that. Unfortunately, we didn't have that luxury, and so we had to design and build the workflow as we were executing it, which sometimes meant we didn't do things in quite the best way, and we've learned from that. If we want excuses, then actually, well, we were in the middle of a program that was going to take us 6 to 12 months to put in place these workflows and get them fully optimized. But unfortunately, the virus struck us before we were ready, and so we had to we had to work this out as we went along. So this phase 1 workflow is described in this slide and in this report that's on this URL here, which describes the methodology in quite some detail. The individual steps we're going to describe in the next few slides, so I won't dwell on this slide. Essentially, the workflow can be broken down into these stages. The first stage is the hit expansion and the ligand preparation. So that's preparing the ligands for docking. The second step is preparing the protein for docking. The third stage is doing the docking itself and generating the docked poses. The fourth stage is scoring and filtering those docked poses. And the final stage is analyzing the data and trying to make some sense out of it. So in more detail on these, the first step, and this is one of the more interesting ones, because some of this is a fairly standard virtual screening workflow involving docking, but the beginning and the end bits are quite novel. So the first step is, we could really phrase the question, if you were given 17 fragment hit structures, so that's 17 molecules, how would you find an extended list of molecules that you wanted to put into your virtual screen? So you might think of doing similarity search. It's a well-established way of finding similar chemical structures. But that doesn't really work in this case, because similarity search doesn't work very well for small molecules. It doesn't work that well for big molecules, but it's for small molecules like fragment hits, which have very few atoms, very few bits in the fingerprint get set, and so you don't get a good signal. You can identify very similar molecules, but as soon as you get away from very similar ones, you quickly get into the noise, and you can't distinguish real similar ones from ones that are really just in the noise. And it isn't chemically intuitive. If you put those structures in front of a chemist, they might question whether they really are similar. So similarity search doesn't really work. Substructure search might be your other idea, and that is possible. You can definitely identify molecules in databases by doing substructure searches, but the problem here is you need to be quite a skilled medicinal chemist to do it, and it takes quite a bit of time. You've got to run lots of different searches for each structure, try them different ways, and it takes a lot of time. It's hard work to do even for one molecule. You certainly would be quite an effort to do it for 17 molecules or even more that we have now. So coming to our rescue is something called the Fragment network, and that's what we use, and we'll describe here. So the Fragment network is something that was conceived by Aztecs Pharmaceuticals, and it was published in 2017. And what it uses is fragmentation of molecules, and it builds up a big graph network of molecules and their fragments. And you can traverse that graph network and identify similar structures. And this is indicated in this graphic in the top right corner, where we start with this particular molecule, and you can identify various variants of that that are very obvious analogs. And what the Fragment network lets you do is to expand out one of those hits or all of those hits and generate these candidate molecules that we're going to include in our docking runs. And it does that in a way that is really quite chemically intuitive. When a chemist looks at these, they say, yeah, they are similar, which, as I said, doesn't always happen if you're using fingerprint-based similarity searches. So underneath this is a database of compounds available from Enamine, ChemSpace, and Molport. And those molecules are available either off the shelf or with a very clear synthetic path, so they can be synthesized quickly and relatively cheaply. And we use the API that sits underneath the Fragment search application to do that search. So using that API, you can almost instantly, just within 10 seconds or so, expand out those 17 initial hits to get a very large number of candidate molecules. And in the work I'm describing here, those 17 hits were expanded out to 42,000 molecules. I say with just one API call. You didn't have to do a great deal of thinking. Makes it very easy to identify molecules, candidate molecules to screen. And in the later stages of the work, where we have 22 hits, we're using slightly more aggressive expansion parameters, and we're getting 85,000 molecules out. So it's a very nice way of identifying candidates for this sort of study. There's more information in the references that are in the slide notes if anyone wants some more on this. So we have those expanded molecules, those 42,000 in this case. And what we then do is prepare them for docking. And for that, what we do is we enumerate charges of those molecules, because they can have different charge states, and sometimes a bound molecule might not be in its neutral state. And so what we do is we enumerate out those different potential charge states using a tool called dimorphite, which is based on RDKit. It's basically a rules-based enumerator of molecules. And then for those enumerated molecules, we then generate an initial 3D structure using OpenBabel. So the process would start with one molecule here, one of our 42,000. We enumerate out the different charge states, and then we generate a 3D molecule for that, which is the starting point for the docking. That can be done in Galaxy. We've got tools in Galaxy for doing both of those steps. The enumerate charges one, that should be charges, not changes, was created as part of this work. So that's our ligands ready for docking. The next step is to prepare the protein, and that we take a relatively simplistic technique here, which seems to work fine for us. We prepare the protein with OpenBabel, converting it into the mol2 format that we need and protonating it at pH 7.4. What that does is leave the active site cysteine and cis145 and his41 in their neutral ground state. There's quite a lot of discussion we could have around that, but it seems for the purpose of docking to get inhibitors, that's probably the right way to go. The active state is where the cysteine is deprotonated, so it's the thiolate, and the histidine is protonated, so it's a zwitterion conformation. But we think that docking in the ground state is probably the better solution. But that's to some extent an open question. So we've now got our proteins and our ligands ready for docking, so we can perform the docking. That's done with a tool called RDoc that was also created and enhanced as part of this work. And what the RDoc tool will do is generate a number of docking poses for each of those candidate molecules. We typically generate 24 poses for each one. And so what that generates, so the enumerated candidate molecules is 159,000. We've got 17 protein structures, so we dock each of those candidates into each of those protein structures, and we generate 25 poses, which means we end up with 67.5 million poses around, there or thereabouts. And then the final step in the workflow, in the Galaxy workflow at least, is to apply some additional scoring functions to those dock poses. Now, you might think of using docking as the screen, but the issue with docking is it's generally recognized nowadays to be quite good at giving you poses, but it's pretty bad at predicting the binding score for those poses or the affinity. And that's particularly the case for small molecules like fragments. And so docking is not really a good way of filtering those poses, but we treat it as a very good way of generating those poses. So we're treating it primarily as a hypothesis generator, and we're not using it as a filter. Instead, we use some novel scoring functions that have been developed at Oxford University help us do the scoring and the filtering. One called Sucos, which is a shape and feature overlay, which basically answers the question: how does my docking pose align to those original fragment hits? which is useful information to have, because those fragment hits are trying to tell us something about how things bind to the protein. And TransFS is completely different. It's a deep learning algorithm that tries to learn the interactions of the ligand with the protein. Typically, deep learning algorithms that are trying to distinguish actives and non-actives, they very often end up learning the ligand, not learning the interactions. But what TransFS does is do a quite smart trick to try to force it to learn the interaction, not the ligand. It uses toolkits called Nina and libmol-grib, and those require GPUs. And so these calculations are run on GPU nodes on Galaxy. Initially, we had some problems with the GPU nodes. We didn't have many of them, and some of them were unreliable. And so we did have some problems running those initially. But now that both aspects of that are solved, we've now got a larger number of GPUs, and they seem to be behaving much more reliably. And of course, this is a new tool that was added to Galaxy as part of this project. It's a quite complex tool, and it took quite a bit of effort to get that running. And thanks to Bjorn Gruning for helping with that. He did a lot of work on that in that area. So that's our virtual screening complete. We now have a very large number of poses, and we have some scores associated with them, and so we want to analyze that data. So that analysis is ongoing. We've done it for the 17,000 for the 17 original hits. We're working through the later stages of the workflow, and the way we're doing this, what we found most productive is to use Jupyter notebooks for that. And so we have a number of notebooks that are listed here that if you want to go and look at them, where we look at different strategies for how to analyze and filter the data, with the overall aim of trying to identify the best candidates in there that we want to go and purchase and then test. So I won't dwell on this because we don't have time, but there's a lot of information in those notebooks. And I also point out, of course, you can run Jupyter notebooks in Galaxy itself if you use the live.galaxy.eu site. So you could potentially do all of this in Galaxy as well. So I think that's the end of my part of it. I'll just finish by saying what we're currently doing and what we're planning on doing, and then hand over to Simon. So currently we're in the process of analysing that data, and we're trying to secure funding for compound purchase. This work, we need to buy a significant number of compounds. We need quite a large budget for this, and we're in the process of trying to secure that. And I think we're close to doing that. And the expectation is we're going to be ordering compounds in the next 2 weeks, which will then be tested both in fragment screens and in biological assays. At the same time, as mentioned before, we're improving the workflow, trying to make it more rigorous, more efficient, and lots of other improvements. And we're evaluating additional scoring tools that will help us to analyze and filter the data. More in the future, there are more fragment structures coming off the diamond beamline, so we'll be incorporating those. And we'll be extending the workflow to incorporate some molecular dynamics-based techniques to generate some more sophisticated scoring functions. And of course, now that we have this workflow working and reasonably streamlined, we'll be able to apply it to other fragment screening hits that are being done at Diamond as well. So that's what we're doing at the moment. I will finish now and hand over to Simon.
So Tim has given an overview of the workflow in a general sense. What I'd like to do is talk a bit about it from the Galaxy point of view, how it actually works to run such a workflow through Galaxy. So all of this analysis was done on usegalaxy.eu, which is the main European Galaxy server, and the jobs were sent to around 10,000 CPUs, so a very large compute resource, as well as a few GPUs for the TransFS docking, as Tim mentioned. And these were provided by DENBI, the German Network for Bioinformatics Infrastructure, as well as STFC, and some HPC resources from the University of Freiburg. And there will be a session actually in a couple of weeks, which is devoted entirely to Galaxy infrastructure and how the Pulsar network can be used to distribute jobs across a huge network of compute resources. So in this case, across the whole continent of Europe. So just to give that a bit of a plug. But the main thing that I want to do is give a small practical demonstration of how to run these workflows and to make you aware of these 3 links. So the first one is just the link to cheminformatics.usegalaxy.eu, which is, so to speak, our cheminformatics flavor of the main Galaxy server. Then our documentation for this project, which is quite extensive, should provide a lot of details that we can't provide in a presentation like this, and also a small tutorial which describes the workflow and hopefully help you to get to grips with the different tools and workflows being used. So let's start with the documentation. Tim showed, Tim showed a screenshot of this already, but basically it's a quite detailed description of all the different workflows. Links also to the workflows in Galaxy, and probably most importantly, links to the raw data. So for each of the fragment hits, which Tim mentioned, the workflows run in a separate Galaxy history, and then links are provided to all of the Galaxy histories. So an example would be, for example, this history. So for each of the steps which Tim mentioned, then you can see corresponding data sets or collections. So the enumerated candidates, fragment hits, the active site definition, transf scoring, SUCOS scoring. Then the next thing I wanted to show you briefly is the website of the Galaxy Training Network. So in case you didn't know, Galaxy has quite an extensive collection of tutorials and training to accompany the different tools that we provide. So we have quite a few devoted to computational chemistry, and one which we wrote recently, which corresponds to this workflow. So the idea behind it is to perform the workflow which Tim's described on a small scale. So obviously you can't perform this kind of workflow on 40,000 different molecules as a tutorial, but you can do it on a very small scale using a 100 molecules. So this tutorial describes for each step a hands-on box to tell you which parameters to use, different tools, what files to upload, also a description of all the differences between the tutorial and the original work, which Tim has described, and then some other bits like question and answers, details of different parameters for the tools and comments. So what I'll try to do now is to actually do a bit of a live demonstration. So we start, as Tim said, with a list of molecules in SMILES format. So in this case, just 100 molecules. In the actual workflow, it would be more like 40,000. And then there are some preliminary steps to prepare. So these I won't go through in detail, but the first one would be to enumerate the charges, as Tim said. So we have a tool for this, very simple tool. We simply give in the minimum and maximum pHs, and then it generates all the different possible all the different possible charge forms for the input molecules, again in SMILES format. So we end up with about 400 molecules. So an average of 3 or 4 charge forms are produced for each molecule. And then for each of these enumerated charge forms, then we then convert to a 3-dimensional format in the SDF format. So if you're aware of basic cheminformatics files formats, then you'll know that SDF can be used for representing not only the bonds and the connections between atoms, but also the 3-dimensional structure. So for this, we simply select SDF here, and there's a lot of different options, but the key one is generate 3D coordinates. So we select this, and then we run the job, which will give us a list of about 400 different compounds with their three-dimensional coordinates. Then the next step is to split this file into a collection. So collections are a slightly more advanced feature of Galaxy. Basically, they allow you to introduce some structure into the history. So, for example, in this case, the initial file with 400 molecules is divided into 10 different files, each with about 40 molecules in. And the reason to do this is to make the best possible use of those 10,000 CPUs, which we had at our disposal. So it meant that we could run multiple docking jobs side by side, rather than having a single massive job, which would have taken a long time to execute and wouldn't have made good use of the resources. So the first step that I'll actually try and do live is the docking itself, because that's really the most interesting one. So for the receptor, then we select dataset 3, receptor in Mol2 format, the active site, receptor active site, which is dataset 4. Then for ligands, we choose a dataset collection. This is this 117. So as it says here, separate jobs will be triggered for each of the datasets within the collection. So we'll be running, in effect, 10 docking jobs at the same time. Then various other parameters, which we can leave more or less as they are. I'll set the number of dockings to 2, which is quite low, but just for the sake of getting the jobs completed in a reasonable time. We don't need to filter the results in this case, so we can just get things going. So it creates a collection here with 10 different datasets, which should hopefully start to run fairly soon. So we'll see them changing colour from grey to this sort of beige colour, and then eventually to green when they're complete. So it looks like things are going quite quickly currently. So I'll go straight on to the next step, which is to convert this collection of datasets back into a single dataset. So we select here this collection dataset, the collection 18, with the collapse collection tool, and we can just start it. Okay, so this may take a bit of time. So, I mean, what I'd like to finish showing you is the scoring. So, as Tim said, there's 2 different scoring methods: the transfs post scoring. The transfer scoring. So, as Tim said, this is a method which uses deep learning to score the poses created by the docking. So, sorry, I see that some data sets are missing here, so I'll just add them quickly to the history. Okay, so for the receptor, then we take the PDB file, which I just moved to this history. For the ligands, I will take the one of the docked files. So something seems to have gone wrong here, because the files from the docking are actually empty. So again, I'll move one of the data sets from a history that I calculated beforehand. Okay, let's see if this works. Okay, perfect. So we have our collection with 700 poses. Distance to waters is the number of is the cutoff distance for the water molecules in the crystal structure which we consider, and then there are various different options you can choose for the prediction. Then we simply start the job. The Sucos score, as Tim said, is completely different to the TransFS score. The idea is to actually to benchmark our docking poses against the experimental data. So the poses that we generate should be seen as a kind of hypothesis, and by comparison with the original fragments, we can then say if those hypotheses are actually similar to the experimental data or not. So for this, we have to choose the ligands to be scored. So we take this output of the TransFS job. We then, of course, have to compare against the structures of the original fragment hits. So here we choose this hits SDF data set. Filters we can leave blank, and then we again start the job. So when these two jobs are complete, then we'll have an SDF file with scores for both the TransFS and the Sucos scoring included as values in the SDF file. One thing as well that I'd quite like to show you while this is completing is how this entire workflow can be run at a single click effectively. So what I've shown you so far is how to run individual tools one after another, but for a kind of high throughput analysis, then you really want to be able to run an entire workflow, and Galaxy allows you to do this. To chain tools together into workflows, and then to run them as if they were a single tool. So under the list of workflows here, then I click on Run, and then it offers various different options, just as if it were a tool. So I can select yes to send results to a new history. The receptor, again, the receptor in mol2 format for the active site, receptor active site for the candidate ligands, this collection, and then for the reference molecules, again, the SDF hits, and then I simply press run workflow. And then the entire workflow is run as if it were a single tool. So that brings the demonstration more or less to an end. So if you imagine that we have 17 or 22 different fragment hits, and we run the workflow on each of those, then it creates a whole lot of different histories. So you can see this shown in a screenshot. So each of these columns represents a different history, and you can see data sets gradually changing from green to beige, or these data sets green, and gradually the data sets turning from grey to beige to green. And Galaxy offers different types of ways to visualize data, either in the form of viewers of the protein structure, like this NGL viewer, or different ways to extract values from the SDF file and to plot them using the various plotting tools available in Galaxy. Right. Okay. So that brings us, I think, more or less to a close. We'll return to the questions in a second, but first of all, many thanks to various people who were involved in this project. So first of all, Frank's colleagues at both Diamond and at the University of Oxford, and Tim's colleagues and my colleagues from Informatics Matters and the European Galaxy team. And thanks also to all these organisations who supported us.